Hello everyone, and thank you for tuning into my talk. My name is Anna Vlot, and I'm a third-year PhD student in the OLA Lab for Computational Regulatory Genomics at the MDC in Berlin. And today I will be talking about a tool I developed for single-cell marker identification by enrichment scoring, which we somewhat creatively abbreviated to SEMITONES. So why do we need SEMITONES? The fundamental characteristic of SEMITONES is that unlike the commonly used marker gene identification by differential expression testing, it does not rely on prior clustering of cells. We developed this approach because clustering of single-cell omics data is non-trivial, particularly for differentiating cells. So let me briefly illustrate this using some graphics inspired by the haematopoietic system. So I'm quite sure we've all seen an image like this that illustrates how a common progenitor cell can follow different paths through this landscape and end up committed to a specific fate. This process can be viewed as continuous, meaning that cells gradually change along the trajectories they can take to become terminally differentiated cells. If we look at these terminally differentiated cells in some 2D projection of whatever omics measurement we have, we expect to see something like this, where groups of cells that have the same identity cluster together. I hope you can appreciate that identifying cluster boundaries here is feasible, and differential expression testing between these clusters might be a reasonable approach for marker gene identification. However, if we look at the complete haematopoietic system, we see a different story as I illustrate here. Although we can still identify the final cell types, we also observe many possible intermediate cell states between the hematopoietic stem cells and the ultimate cell types. If we want to cluster these cells into groups of cells with the same identity, it is far less clear where the cluster boundaries should be. So these issues were the motivation for the development of SEMITONES. So let me explain the method. So let's stay with this 2D representation of a differentiating system, and let's assume we are looking at gene expression data. If we're interested in marker genes for this highlighted region, we select a cell to represent this neighborhood, which we will refer to as a reference cell. Next, we define a marker gene as a gene that is only expressed in this reference cell and cells similar to the reference cell, like the expression pattern illustrated here in orange. From this it follows that there exists a linear relationship between the similarity to the reference cell and the expression profile of a marker gene. As such, we may use simple linear regression to infer the regression coefficient that quantifies the strength of this linear relationship. This regression coefficient is then interpreted as an enrichment score that quantifies how likely it is that higher gene expression values are found at the top of a list of cells, ranked by their similarity to the reference cell, than at the bottom of said list. Ultimately, SEMITONES assigns high positive enrichment scores to genes that are only expressed in the cell neighborhood, or marker genes, scores at around 0 to uninformative genes, and low negative enrichment scores to genes whose lack of expression is exclusive to the reference cell neighborhood. These genes might also be highly informative as absence markers. For the identification of highly specific marker genes, it often suffices to simply look at some of the highest scoring genes. However, there exist situations in which significance testing is desired, and for this, SEMITONES computes a null distribution of scores for 100 times permuted expression vectors. In practice, this distribution represents the expected enrichment scores for randomly expressed genes. Significance is then declared at a user-defined number of standard deviations away from the mean of this null distribution. So now that we've covered the motivation and the method, I will show several applications of SEMITONES and compare SEMITONES to the current most popular and alternative cluster-independent marker gene identification methods. So what we see here is a two-dimensional embedding of single-cell RNA sequencing data of healthy human hematopoiesis that was published at the end of 2019 by the Greenleaf Lab. It consists of roughly 35,000 cells from samples of stem cell-enriched bone marrow, and peripheral blood, which means that the whole hematopoietic development from stem cell to fully differentiated blood cells was captured, making it a suitable data set to explore the usability of SEMITONES. The single-cell RNA-seq data was obtained using a site-seq procedure, so a ground truth on cell states is also available from the observation of cell surface markers. The goal was to apply SEMITONES to identify marker genes for all cell states in this system. So to do this, we need a set of reference cells that is representative of this whole population. In the SEMITONES package, we provide several ways to do this, but in this case, we simply took the cell whose average dissimilarity to all cells in the sample is maximal, and then iteratively selected the most dissimilar cell to the previously selected cell, resulting in the selection presented here. Given these reference cells, we then compute enrichment scores for all genes in each reference cell using semitones. If we then inspect the 5 to 10 most enriched genes for each reference cell, we can annotate all these reference cells based on these genes. These annotations agree with those in the original data publication, confirming that semitones identify cell identity markers. We also annotated additional cell type subsets, for example for monocytes and T cells, and more intermediate states of B cell development. The highly granular annotation of cell subsets is possible due to the identification of highly specific and overlapping markers, as I illustrate here using the top scoring marker genes along the B cell developmental trajectory. DTX1, for example, is highly specific to transitional B cells, while VPB1 is known to be most highly expressed in pre-B cells and is most enriched there, but it's also expressed in common lymphoid progenitors, pro-B cells and transitional B cells. It is the relative enrichment of these genes in a particular cell neighborhood that allows for specific cell state annotations. Now, this evaluation of marker gene retrieval is rather qualitative, so we also sought to quantify the marker gene retrieval capabilities of semitones. So for this, we checked how many of the markers we found for the healthy hematopoiesis data are found in the cell marker database, which is a database containing marker genes from single-cell RNA-seq experiments. We subsetted this database to only contain markers for human hematopoietic cells, and the marker genes were divided into high and low confidence markers, where high confidence markers are supported by 3 or more sources, and low confidence markers by 1 or 2 sources. Then we assign each gene its highest rank in any of the reference cell and divide the genes into 25 rank-spaced bins, as plotted on the x-axis of this plot. Next, we compute the frequency with which genes in each bin are present in the cell marker database, as displayed on the y-axis. From this visual, we can take away that high-ranking genes are more likely to be high-confidence markers than lower-ranking genes. And for example, in the top 25 rank bin, just over 20% of genes are high confidence markers and a total of 73% are present in the cell marker database. When I evaluate the remaining 27% of genes further, I find evidence of marker gene status from either other databases like GeneVisible or the literature for another 17% of genes, increasing the total percentage of genes for which evidence of marker gene status was found to 90%. Then here, I show the expression profile of genes with a rank between 1 and 10 in the remaining 10% of genes in the top bin. From this, I hope you can appreciate that many of these genes seem to have neighborhood-specific enrichment of expression and are therefore potential novel marker genes. So the next thing I wish to show you is how semitones compares to 2 alternative approaches. One is single-cellHaystack, a method for clustering independent differential expression testing, and the other is arguably the most popular method for marker gene identification, namely Seurat's default Wilkinson rank sum testing between clusters. For this, we provide Seurat with the surface protein marker informed clusters from the original data publication to make sure we use high quality clusters and assess Seurat's performance in a best case scenario. So in this figure, I plot the gene rank according to semitones on the y-axis and the rank according to single-cellHaystack and Seurat on the x-axis. The first apparent observation is the higher agreement between semitones and Seurat than semitones and single-cellHaystack, but we also see disagreements in rank between semitones and Seurat, both for genes ranking in the top for semitones and the bottom for Seurat and vice versa. Unfortunately, I don't have time to go through the characteristics of all disagreements, but I do want to provide an example of the type of gene only identified by semitones, using this example of a pre-dendritic enriched gene, IGSF6. This gene is only enriched in a small number of cells and not accurately captured by a specific cluster. Rather, it is only expressed in a subset of cells in the granulocyte and monocyte progenitor cluster here. Now, of course, all 3 of these methods identify biologically relevant and informative genes. But these results do show that semitones can identify highly specific markers that are not identified by alternative approaches. Again, this comparison is qualitative, so we set out to compare semitones to alternative approaches in a quantitative manner too. For this we use simulated data obtained using Splatter for 1,000 to 10,000 cells, and Splatter provides a ground truth on differential expression for each simulated gene, allowing us to compute ROC scores to compare semitones to single-cell-Haystack, monocole 3, and several differential expression testing methods using Seurat's default gene filtering or without any filtering. So in this graph on the left, we can see that semitones achieves the highest median ROC score when using an optimal similarity metric to compute the similarity to the reference cell. But even when semitones uses a suboptimal similarity metric, its performance is on par with differential expression testing without prior filtering and monocole 3. However, if we look at the plot on the right, we see that semitones does so in up to 30 minutes, while monocole takes up to almost an hour, and differential expression testing up to almost a day. So now that I have elaborated on some results regarding semitones accuracy for marker gene identification, I want to show a few more application examples that illustrate the flexibility of the framework. So the first application I will show is the application to chromatin accessibility profiles using single-cell attack-seq data of healthy human hematopoiesis from the same publication by the Greenleaf Lab, which also contains single-cell attack-seq data for roughly 35,000 cells. The application procedure is virtually identical, and we first select 75 reference cells from the sample, and then we compute enrichment score for all peaks in this dataset. If we look at the top enriched peaks, we very rarely identify known regulatory regions, like this monocyte enhancer, but generally speaking, the function and cell type specificity of these genomic regions is poorly annotated. So in order to annotate our reference cells, we pass the genomic regions with a significant positive enrichment score to GRADE, which is a tool to analyze the annotations of genes close to the provided genomic regions. Reference cells were then annotated based on the functions provided by GRADE, and these annotations again correspond broadly to those provided in the original data publication, which were based on an integrative analysis of single-cell RNA-seq and single-cell attack-seq data. So from this we conclude that genomic regions identified by semitones contain cell type specific information. Besides selectively accessible regions, biologically meaningful insights can also be derived from selectively inaccessible regions. And here we see the percentage of regions that have a certain genomic annotation from HOMA or are annotated as enhancers in Phantom 5 for all significant regions in purple, selectively accessible regions in green, and selectively inaccessible regions in orange. A particularly interesting observation here is the relatively large percentage of inaccessible regions that are promoters. If we look at the function of the genes corresponding to these promoters, we find some sensible biological functions. For example, in NK cells, we see that promoters of genes involved in the downregulation of IL-8 production are inaccessible. And likewise, promoters of genes involved in T-cell lineage commitment are inaccessible in transitional B and pre-dendritic cells. Taken together, these results signal that semitones identify cell state-specific cis-regulatory regions from single-cell attack-seq data. Then finally, given an increasing popularity of spatial transcriptomics methods, I will show you an application of semitones for the identification of spatially restricted genes. And for this I use 10x Visium data of the mouse brain, which provides transcriptional profiles linked to a 2D location in a histological image of the anterior brain. Since the 2D location of the transcriptional profile is provided, we simply fit a 2D grid over these coordinates and select locations at the lattice points of this grid as a reference location for enrichment scoring. By applying semitones to all genes and all reference locations, we retrieve known spatially restricted markers, like FTH1 for the fiber tracts, GNG4 for the main olfactory bulb, and CAP-B7 for the hippocampus. But we also identify a Riken gene, 5015-010-RIC, as a more selective marker for the choroid plexus than the more commonly described TTR gene. Using the marker GNG4 for the main olfactory bulb, I want to show you one final application of semitones, and that is the use for location annotation given a marker gene. In the image on the left here, we can clearly see the main olfactory bulb in dark purple, and by computing the enrichment score of GNG4 for all locations, we get the distribution of enrichment scores shown in the middle plot. Subsequent significance testing then allows for the annotation of locations as belonging to the main olfactory bulb if their enrichment scores exceed a certain threshold. On the right, we see how we recapitulate the shape of the main olfactory bulb using this approach. So although the applicability of this approach is more easily evaluated using spatial transcriptomics data, the approach can also be used for cell annotation in single-cell RNA-seq data, as was suggested and performed by Che Wei in the lab for the annotation of an Arabidopsis root atlas, which is available on bioRxiv. So to summarize what I've taught you so far, semitones is a tool for the cluster-independent identification of highly selective marker genes. Semitones can be applied to diverse omics data types, including single-cell RNA-seq, single-cell attack-seq, and spatial transcriptomics data. And finally, besides the identification of marker genes, semitones can also be used to annotate individual cells or locations based on known marker genes. Now, two further applications of semitones that I did not talk about today are the use of semitones for the construction of co-enrichment graphs to identify regulators of cell identity, and its application as an alternative to highly variable gene selection. In case you're interested in these applications, please drop by my poster later, read the preprint of semitones on bioRxiv, or dive straight into the source code on GitHub. So finally, I want to thank all the members of the OLA Lab, in particularly Che Wei, for inspiring discussions regarding the applications of semitones, and my supervisors Uwe and Setare for their support, and then Hybrids for funding. And then of course I want to thank all of you for your attention, and I hope to see you live shortly to answer any questions that you might have.